Awesome. Thanks, Ben. Um, so yeah, this is let you read the title a second. See my Twitter handle, all that. So we are in Holland, Michigan, which is here on the west coast of Michigan. So I get to watch sunsets over Lake Michigan. We're about 3.5 hours around the lake from Chicago, about 3.5 hours across the state from Detroit. Settled by Dutch immigrants in the mid 1800s, and they brought Dutch windmills and Dutch tulips with them. The town and the college is about 6 miles inland from Lake Michigan at the point of this little inland 6 mile long, half-mile wide lake. And back in 2004, an assistant professor could afford to buy a house here about a quarter mile as the crow flies from the beach, and about a mile walk to this state park and a mile walk in the other direction to a really gorgeous county park. But you didn't come for a sightseeing tour. I'll tell you a little bit about our chemistry. I'd start with some introductions. I know we've got a diverse audience on, and so not even all chemists. So we'll walk through the title as a way to do some introductions. First of all, when I say organic, some of you guys think of this, and that's not what I mean by organic. I mean the chemistry of carbon. That's what an organic chemist means when they talk to you about organic chemistry. And so literally every molecule you'll see in this talk comes from those 9 elements. Now, we got some helping elements along the way, so another dozen or so here, but still less than about 25% of the periodic table to do almost all the chemistry of life, the chemistry of synthetic plastics, of pharmaceuticals. When we talk about organic chemistry, that's the kind of stuff we're talking about. When I talk about research, I mean the same kind of research that happens at USC and MIT and the ETH. We compete for the same grants. We publish in the same journals. We just do it at a slower pace and need to have topics that are just a little bit more niche because we do it all with undergrads. So the JGG group has never consisted of anybody but me and about a half dozen undergrads at a time, usually 4, 5, or 6 is kind of normal. Ben already told you a little bit about Hope College, so I won't belabor that. When we talk about synthesis, we mean making molecules. And so one kind of organic chemist is a synthetic organic chemist. That is not me. I've got a colleague that says all chemists are either makers or measurers. I am absolutely a measurer. Unfortunately, I've never been able to buy anything that I want to measure. And so we spend a lot of our time making molecules. When we talk about photochemistry, we talk about the end products, in my case. Sometimes you can use it synthetically, but the end products are molecules that interact with light. So you can have them do chemical reactions where light is one of your reagents. And when we talk about computation, we're not talking about this stuff here that I'm not very good at, which is why I like teaching organic chemistry better than general chemistry, as it's the least mathy of the chemistries. When I talk computation, I don't mean this, I mean that. I mean supercomputers and having them simulate molecules for us. And actually, this is also something that is not really up my alley. I came to Hope College never having done a computation in my life. We've got some really great computational resources here, and I've got colleagues that I can that can kind of mentor me in learning to use this as a tool. All right, so that's the introduction to my title. A little bit of introduction to me, since you're not even seeing me on my screen. This is a slightly younger, better looking me, and some things that I like. I like taking my Jeep up into the dunes around Lake Michigan. I like sunsets over Lake Michigan. I like lifting weights, and that's my beautiful wife. And she's an amazing deadlifter, better than I am. I like being a karate dad and a dance dad. And my kids even got me into some karate of my own. I like coffee and beer that looks like coffee, especially if it's spent some time in a bourbon barrel, or just give me the bourbon. And I absolutely love chemistry. Only thing I love more than chemistry are these folks, and maybe this guy, our pandemic puppy. But I really love chemistry, and it started in New Jersey, and this is the part of New Jersey that I grew up in, Hunterdon County. It is pretty and rural, and I spent my first 8 years on a horse farm. But I did go to high school still in that same rural setting, directly across the street from Exxon, now ExxonMobil's corporate research labs. While I was there, I took AP chemistry. I knew a lot of chemists and chemical engineers, and in early 1990s, the chemists and the chemical engineers said, go be a chemical engineer. So I went to Virginia Tech thinking that's what I wanted to do. But that AP chemistry course, the chemists talked to me about this great organic class that I could take as a freshman, only 75 students, not 300, and taught by this master teacher, and how great it would be. And the engineers told me that it was great that I had AP chemistry because then I could take both Fortran and Pascal. That's maybe dating me a little bit, but 2 programming languages. I asked, when do I get to take organic? And they said, oh God, hopefully not until your junior year. And so that seemed a little too far away for me, and I became a chemist. After I graduated with my BS and my MS from Virginia Tech, I went up to the University of Rochester, where I learned to appreciate photo-induced charge transfer as a tool. And so using light to create cation radicals that did cool material science things. So we worked on projects related to volume holographic data storage with the folks at Kodak Research Labs, which was still a kind of a Bell Labs type place back then. Really cutting edge science that has since ceased to be the case. But back in the late 90s, Kodak and Rochester were a fantastic place to be. I did a short postdoc at Vanderbilt where I tried applying my mechanistic organic chemistry to more biological problems and really realized that I like materials problems better, but I enjoyed the music scene for a year too. Before I came to Hope College in 2004, this is my first research group, right? Photochemists actually play in the dark a lot because we don't want the light to do things with our molecules we don't want them to do. So we've got red windows and drapes and curtains and stuff. And I mostly teach organic chemistry. And before the pandemic, I did it all on chalkboards. I've since learned to teach on this less ideal medium, but this is what's near and dear to my heart. I have had 2 sabbaticals. One, I worked with a family of dyes in the Arizona State University Center for Bio-Inspired Solar Fuels. And then the other, I just went down the road to the Michigan State University's Bioeconomy Institute in their organic energy storage lab and worked on additives for lithium ion batteries and redox flow battery analytes and catholytes. But at Hope College, the majority of my time was spent on a project that I'm going to only give you a single slide on. It was what we called photochromic photooxidants. So photochromes are things like the dyes and transitions lenses, right? You go outside in the sunlight and they become colored. You come back inside and both thermally and normally with visible light, they get driven back to a colorless form. So we studied a family of photochromes called the pyrimidine spirohexadienones for my first 12 or 14 years at Hope College that did not go back from the colored long wavelength form to the colorless short wavelength form photochemically. It was a purely slow thermal fade. So they would have made really crappy transitions lenses. You'd go outside, you'd get sunglasses, you'd come back inside, you'd wait 6 or 8 hours and you could see again. We weren't trying to make transitions lenses. We were trying to leave that photochemical channel open. And so had there been a unimolecular photochemical fade, we wouldn't have been able to do what we wanted to do. And so what we were trying to do was make a system where the long wavelength form of the dye was a good photooxidant, able to do those kind of photo-induced charge transfers that I had done at Rochester and with Kodak. And from the long wavelength form. And so essentially adding an extra level of gating to photo-induced charge transfer. First, you had to use UV light to convert to the long wavelength form. Then you had to use long wavelength visible light to do the photo-induced charge transfer reaction with some donor molecule that you wanted to oxidize. And so we had some pretty good success with this, published several papers on this, but also realized that I could tell you what would make something a better or worse photooxidant. That is, I could tell you what would make something more or less reducible, but I couldn't tell you by how much. And so this was my first foray into computational chemistry. And actually, we did some really naive things because we got a grant internally. We had this really nice computational resource and it wasn't getting enough use. And so they put out a call for proposals to use it more, and it would fund some students to use the computational cluster. And we said, well, we're going to compute for some reduction potentials. And they laid out a plan of why. What we didn't lay out was how, because I figured we'd go to the library and find out how. And at the time, it turned out there weren't great ways to compute reduction potentials of molecules about this size in a reasonable time frame. And so the student and I made some just drastic approximations and tried really overly simplistic things. And it worked out beautifully due to some nice cancellation of errors and being able to correlate to experiment. Rather than computing an actual value, we could just compute something that was easy to compute. And then we could correlate it to known reduction potentials. And then we could use that correlation to predict the unknown reduction potentials of molecules that we could only compute but not measure. So that worked out well. It was actually my first paper at Hope College was not in the Journal of Organic Chemistry, but in the Journal of Physical Chemistry. And it was about this computational stuff that I had never done a computation before I set foot on campus. And now today, my group is probably about 20% computational, about 80% experimental. And just as this project was starting to get to the point that the synthesis was getting as challenging as it was going to get, these dyes can open in either of two directions, right? I show you here it opening towards the ring with the nitrogen in it. It could also open away. Things that we could do to make this dye more reducible started to increase the chances computationally that they would open the wrong way. So we're starting to hit the point of diminishing returns. And I happened to have a colleague in the engineering department, in the mechanical engineer, and really kind of a material scientist, but he's employed by Hope as a mechanical engineer who's interested in photomechanical materials. And we think of these as new modern materials, but nature's been doing it for just about forever, right? So plants are a photomechanical material. And so we'll speed up this video. It's a 24-hour time lapse here that you're going to see in 20 seconds or so, and you're watching pea plants follow the light, right? So things that bend or move in response to light, that's photomechanical. And so this next video here down below is actually in real time. And if you've got a decent sized monitor that you're looking at right now, then this is actually approximately to scale too. This is a thin little piece of plastic that has a laser beam shining on it. And as they switch the polarization of the laser beam from vertically polarized to horizontally polarized, that piece of plastic is bending back and forth in response to light in real time. So that video is not accelerated at all. And this is coming out of the group of a guy named Tim White at the Air Force Research Lab down in Dayton, Ohio, who's really a world expert at this. And my buddy that I told you about, the material scientist who's got a job as a mechanical engineer at Hope College, his name is Matt Smith. And Matt is really into these kind of materials. He had done a postdoc at Air Force Research Lab. And that plastic that you watch flip back and forth there as we change the polarity of the light was based off of something called an azo dye. And so these are dyes that have been known since the 1800s. One called butter yellow was actually invented here in Holland, Michigan. Butter yellow was used to make margarine, which by itself would normally be as white as this screen, look a little bit more like butter in the 1950s. So these have been known for a long time as food dyes, as textile dyes, and more recently for things like photomechanical materials. There's been a real resurgence and interest in them because that azo double bond can switch from a trans form where the ends are opposite each other to a cis form where the ends are near each other. And that's a really big molecular motion. And when we do chemistry, we tend not to think about the fact that our molecules change shape because in fluid solution, it doesn't really matter very much. But the way a photomechanical material works is it couples that molecular shape change ultimately to macroscopic shape change and bulk mechanical motion. It does it by incorporating the dyes into a polymer. It's pretty obvious to me how this would work if it was along the main chain of the polymer or on a crosslinker between two chains. Less obvious, but still true, is even if you just have these dangling off the chain, that molecular shape change can change the shape of the material. This happens right when chains pull each other. If you imagine a trans extended form go into a cis form here, you're going to pull on the ends of that chain. I can maybe help you see a little better if I turn on my laser pointer. And so this microscopic shape change, if we can get these all aligned, either all the materials lined up or we only hit the ones that are lined up the direction of the laser beam, that's why we use the polarized laser beam in that video I showed, then you can get all of these motions pulling in the same direction and either get a 2D contraction or expansion or a 3D bending or unbending like we saw in the video. And right, my engineer buddy will talk about strain gradients and things like that. We don't need to worry about that right now. And the applications for these things are for on-off switches and valves and micromotors or micropositioners. Frankly, these are all things that we know how to do, right? We can do these with mechanical valves and motors and positioners and electricity. This is why the Air Force is interested in this. The other place that's interested in this is NASA, because we're not better than mechanical versions of these, but we are lighter. And so if you want to put it at altitude or in orbit, this is a really neat application. And so people have been studying these for well over a decade now. And it turns out these azo dyes are the kind of current state of the art. They're the best things for this. And azo dyes are great, except for one thing. They absorb from the UV up to maybe the blue-green edge. That's the high energy end of the visible wavelength spectrum and even into the UV, right? This is the kind of the end of the spectrum that will give you a sunburn. This is the end of the spectrum that'll keep your fries warm at McDonald's. So this short wavelength, high energy end of the spectrum comes with some problems. For one thing, pretty much everything absorbs there, right? The one ding that people give organic chemistry is that if you do a good job, you get a white solid, and if you do a bad job, you get a brown oil. That's because things absorb down here in the UV, the violet, the blue. So yes, your dye absorbs there, but so does just about everything else that you're going to put into your material. And right, those sunny beach days, my kids tend to leave some plastic stuff in the backyard, right? Shovels, toys, whatever. And if they leave them there all summer, by the end of the summer, they're bleached, they're faded, they crack, right? That's photodegradation. And like I said, UV gives you a sunburn and blue light's not getting through you. So one of the things that people propose photomechanical materials for is things like transdermal drug release. That's not going to happen with blue light, right? If you or your kid holds a flashlight up to your hand or puts it in your mouth, it's only the red light that gets through. So that's why the community was really excited when a guy named Ivan Abrahamian at Dartmouth published these long wavelength BF2 conjugated azo dyes. And so there's still a nitrogen-nitrogen double bond, still a phenyl ring on one side, but we've got a boron bridge like a Bo-Dippy and a longer conjugated system through what used to be a quinoline ring. And because of that, these things absorb out in the longer wavelength visible and even tail into the near infrared. So the community was super excited about this, including my friend Matt, who comes to me and says, hey, you know how to make dyes. Can you make these dyes? So can we make and study these at Hope? Sure. Turns out the synthesis of these is easier than the pyrimidine spirohexadienones that I spent my first decade plus at Hope at. If there's organic folks in here, right, you make an N oxide and then turn it into a chlorine. This is kind of an awkward two-step process, but it was the best Abrahamian could do. Simple SN2 cyanation reaction. To make this quinolinal acetonitrile, that's sort of a linchpin piece in the synthesis. And then you can take any aniline derivative you want, make a diazonium salt of it. A lot of them, you can even buy the diazonium salts. You couple that with the quinolinal acetonitrile to make this hydrazone. This is what Abrahamian was actually working on, and he had a reaction of this dye that he wanted to do that used BF3 ethyrate, and he discovered these azo dyes by accident. He didn't mean to insert a boron in between. But that is what happens. So now when he optimizes the reaction, he does it with Koenig space, pulls this proton out from between the two nitrogens, puts in a BF2 bridge. You get a 5-membered ring rather than a 6. So it's this nitrogen that you actually coordinate with once you pull the proton out. And these are those azo dyes that I talked to you about of Abrahamian. So yep, we can make them. That's all work done in my lab, but replicating work that Abrahamian had done. And so this is a UV vis spectrum. So these are really dilute solutions of the cis on the trans dye. This is a UV vis spectrum. So it's just really telling you what color something is, the wavelength of light that it absorbs, and how much of that light it absorbs. So the trans form absorbs at slightly long wavelength than the cis form. And you can drive these back and forth. You can shine light over here and turn the trans dye into the cis dye. You can shine light over here where only the cis absorbs and drive it back to trans. And again, we could do this at Hope College. We used a mercury arc lamp with an appropriate long pass filter to make that happen. Another technique that we like to use to characterize things is NMR. NMR is basically like an MRI for molecules. It's really the exact same technology that's being done on your body to look at the protons and water molecules in your body, except we're looking at the protons in organic molecules. And you can see that the cis dye has protons that are different than the trans dye. And we can completely convert the trans to the cis. And we have nice spectroscopic handles. And you see when the dyes are more concentrated, because NMR is a slightly less sensitive technique than UV vis, you get these really beautiful indigo blue and blood red colors. So Abrahamian used the phenyl ring of these dyes to tune the color, right? So he discovered them by accident. So this is the first one he discovered, right? Just like I talked about trans dye, he can convert it to cis dye, he can convert it back to the trans dye. And so this is on the unsubstituted phenyl version. And so he used groups on that phenyl to tune the dye. So if he puts some methoxy there, it goes to longer wavelengths. Here he doesn't get complete reversion, so this hasn't grown all the way back in. There's still a little bit of cis there, but he can convert the methoxy back and forth between trans and cis. If he goes to the dimethyl amino, he pushes to even longer wavelengths, But now the spectral overlap between the cis and the trans is a lot closer. It's a lot harder to get spectral resolution and harder to drive from one to the other. But as he's putting increasingly strong EDGs, electron donating groups, onto the phenyl, he's moving the absorption. And he could get big effects, right? He's purposely trying and he's purposely using really good electron donating groups. He never put anything on the quinoline ring. He always put stuff on the phenyl ring and always at the para position of the phenyl, and he could get huge changes. We frankly, when my buddy approached me, we didn't care about getting huge changes. We were happy to live with this HH 530 nanometer trans 475 cis. That would have been fine. We're not trying to tweak wavelengths. We want to add synthetic handles. And so we also discovered a route that let us get to the chlorination in a single step rather than two steps. We don't have to go through the N oxide. So we shortened the synthesis. And we could put things like halogens at both ends, right? Bromine's not going to be a good electron donating group for tuning the dye. We didn't care. We weren't trying to tune the dye. We just wanted synthetic handles. We put on several other synthetic handles, alkenes, alkynes, alkyl groups. But just to show you where we were coming from, this will suffice. And it turns out we do get some tuning, very little tuning, but maybe more on the quinoline than on the phenyl, right? So I don't have a picture here, but remember R1 was on the quinoline, the big ring. R2 is on the phenyl, the little ring. Abrahamian only played with R2. And so this is compound A is one of Abrahamian's dyes. It's the one I said we would have been happy with. Well, if you put a bromine just at R2, Abrahamian's spot, it's worth 7 nanometers. If you put a bromine on R1 in addition to R2, now we've got a 10 nanometer difference. Or if we compare Abrahamian's H-methoxy, remember that was longer wavelength because methoxy is a good donor. That's 594. We add a bromine at R1. Again, now it's worth not 7, but 13. So the bromine at the para position of the phenyl worth about 7. Bromo 6th position of the quinoline worth 10 to 13. So maybe it's as good or better to tune these dyes on the quinoline moiety. So in the midst of working on these dyes for photomechanical materials with my buddy in engineering, we also got a PRF grant to kind of explore this idea of, well, what if we functionalize on quinoline? What does that do for us? And so, for instance, if 1 methoxy is good, 2 methoxys is better, right? So 6th position methoxy, give you a little bit of a flavor of what organic synthesis looks like, right? There's multiple ways to get to things, right? We can build up the quinoline ring by a thing called a Dubner-Miller synthesis, or we can start with the bromine there and turn it into a methoxy. We're not synthetic chemists. We get crappy yields and live with them. We just want to make the stuff. Well, it turns out that this route is better than that route, except for the fact that the starting materials for A are about 10 times more expensive than the starting materials for B. So when we're feeling cheap, we go route B. Although this summer we've gotten yields for step 1 up to 98%. So maybe we'll do some more route A. Right. And then we told you we were excited. We had this TCCA route to put a chlorine on there. Doesn't work when you've got a methoxy. Methoxy is too strong an electron donor. It wants to chlorinate on the ring by electrophilic aromatic substitution. So we had to devise a more potent benzylic bromination procedure. So we have to beat on this with tons of light, do it really low concentration with a lot of light. So you limited to real small scale reactions, but we can get this to work that we brominate at the right place, and we can turn it into the nitrile, and then we can couple with the hydrazone we want. So now we've made the the diazonin that we want to make the hydrazone where we've got a methoxy on the quinoline and on the phenyl. And so we can make that dye, 2 methoxys instead of 1. Point out just how pretty this chemistry is. This is the hydrazone. So this stuff is that yellow band. This pink band is the cis form of the dye. This indigo blue band is the trans form of the dye. And they elute off the column just like that. Really easy to follow, really pretty chemistry. So not all organic chemistries is browns and whites and yellows. So this is our spectroscopy on that paramethoxy 6-methoxy dye. So added a second methoxy group. And what you'll notice is we didn't change it a whole lot. We went from 594 to the low 600s. So we didn't make big changes. We were sad to see, but we do have the trans dye. We can irradiate out here and drive it all to cis dye. We didn't have the right filter to quite drive it back. And so when we try to drive it back, we reach this photostationary state instead. one of several dyes that we made last summer was one like this. We also, oh, that's the same one. We went hard after this 6-morpholino dye. We went after withdrawing groups as well. I showed you that we had already gotten this one. The slide's a little repetitive. We couldn't get this 6-morpholino dye. There's only a limited number of sequences that you can do it. You got to chlorinate, cyanate, put on the morpholine. No possible combination of those works. If you try to put on the morpholine once the nitrile is there, you hydrolyze the nitrile. If you try and put on the morpholine, you can't halogenate no matter what reagents you use. So we just couldn't make that one work. But the student devised a route to the 4-morpholino that we'll talk about in a minute. And the reason we went hard after morpholino is because it's the best of both worlds. Remember I showed you dimethylamino pushes out to the longest possible wavelength, but has crappy spectral separation. Then the paramethoxy has really good spectral separation, but not quite as long wavelength. Here, the morpholino is sort of the best of both worlds. There's a lot of room out here to irradiate just cis, but we're at the longest possible wavelength. So that's why we tried so hard to get this morpholino compound. And so we can make the quinolinol acetonitrile, we can make the hydrazone, but look at that yield. Because of that yield, the student didn't have enough material last summer to make this dye. So we picked up with this again this summer. And we've learned that position matters, right? So we purposely were working at the 6th position last summer because it's not in direct conjugation with the azo or the phenyl. Lets us tune the quinoline independent of the phenyl. And we saw that with the bromine, hey, that made the same difference either way. Let's work where we don't have the complication of having the two groups in resonance with each other. But now I showed you with that one that we've made and one other one that I haven't shown you, our preliminary results are saying that whether it's something like a bromine that we knew would have a small effect, or something like an acetyl or a methoxy that should have a big effect, we're really seeing minimal effects at the 6th position. So this summer we've moved to the 4 position where we are in direct conjugation with the azo and the phenyl. So we can put electron donors there and hope to have big effects because we're in direct conjugation with the azo. Just like abrahamian was in direct conjugation this way, we can be in direct conjugation from the top. So electron donating groups at the 4 position are an interest, but also electron withdrawing groups at the 4 position. We get this push-pull effect that I've shown the resonance arrows for here. It extends the effective conjugation length. It gives increased single bond character between those two nitrogens. And because of that, we might increase our quantum yield for isomerization, make it more efficient. We might make it fade back from cis to trans more quickly. So we're excited to make these and study them. This summer, we've made a whole bunch of quinolinol acetonitriles and 1 hydrazone. We're 9 weeks into a 10-week summer research, and this is where we're at right now. Hopefully next week, we can make the dye of the 4-methoxy. And during the coming school year, we'll convert all these other quinolinol acetonitriles to their corresponding dyes as well. Right, so this PRF grant was very vague. We outlined electron donating groups and electron withdrawing groups at 4 different positions on the quinoline and a whole bunch of different possibilities. Just the ones that I've cartooned here would be 312 of them. And so we've also used computation to help us select among these. And we thought that that would be really simple, would be that because there was a precedent in Abrahamian's paper. Unfortunately, he only computed 1 dye. He computed just the parent dye. He didn't compute any of the other 6 that he made in the second paper. And it turns out that the 1 dye with no substituents computes very well. All the rest give the exact same result as the one with no substituents. So the computation doesn't take the substituents into account. So fortunately, I've had a really gifted student who does computational modeling using Gaussian implemented through WebMO to simulate spectra. And he's tried a ton of different things. This is the dyes just in their wavelength order, the experimental wavelength absorbance in blue. And these are the 3 best combinations of basis set, and functional, and solvent model that he's been able to come up with for predicting these. And none of them are very good. At first glance, you'd say, well, at least the yellow and the green are kind of close. Turns out the yellow and green are really bad because they bounce around so much. At least the red line is kind of parallel to the blue line. And so it's got some systemic variability, but it actually gets the dyes in the correct order. Always within 1 rank of being in the correct order. And so because of that, you can do a fit and correct the red line to this here orange line that overlaps pretty darn well with the blue line. And so we've just about, the one thing the pandemic didn't slow down was our computational modeling effort. And so we've been able to move this forward. We're actually about ready to submit a manuscript on this, and it's now a useful tool that we can use to inform the synthetic side of the lab as well. So with that, I've got some thank yous to make. This is my group this summer. The computational students just working part-time and was off today when we took this picture at their poster session. But Janie and Ellie are both freshmen that took organic as a freshman like I did. Allie is a sophomore. And so the three of them are in my research group this summer full-time. And then Colin is the part-time computational guy. Some other recent students there. We also have a course-based undergraduate research experience that the second half of our second semester organic lab is an independent synthesis project. There have been about a dozen students that have contributed in the last 2 years to this project through our cure. Right, this all got started with the more materials project with Matt Smith that had several of my students and his, and more of those Chem 256B students. I've had a lot more students on the stuff that I only gave you a single slide about. We've been fortunate to be really quite well funded, active funding here, and previous support. I also need to thank you guys for listening and giving me a chance to share this stuff. This is a fun new medium to be able to do that. So I want to thank Ben for the opportunity for that. Tell you that, put another plug to get some follows on social media. I need to thank these guys as well. They're the only thing in my life more fun than chemistry right now. And I particularly need to put in a plug for this guy. And I don't know if it's appropriate or not, but I'm going to go out on a limb and tell you just a quick story about him. He, kind of on a whim, he's been doing karate now for 4.5 years. He'll be a black belt by the end of August. But he entered a regional tournament in Ohio in February and came in second, third, and fourth place in sparring, in sword, and in forms. And that qualified him to go to nationals in Detroit in June, where he came in second in sparring and fourth in forms. And that was sufficient to qualify him to go to Killarney, Ireland to represent Team USA in the WKC World Championships in October. And not only him, but from our dojang, from our school, there were 13 kids that got to go to nationals, and 8 of them qualified to go to Worlds in Killarney, Ireland, which, as you might imagine, is not cheap. And so absolutely no pressure, but I thought I would throw it out there. My Twitter handle is the same as my Venmo and PayPal handle. If you think that this is a cool thing and you'd like to help these kids get to Killarney, Ireland, you can do that. Put Fusion in the memo line and the money will get to their dojang. So anyway, thank you guys so much for indulging me and let me share kind of my story as well as my science with you guys. It's been fun.
Thank you for that. That's really cool work and a very cool story about your son too. That's really impressive to make it that far. So let's jump into the questions real quick. So I think you'll appreciate this first one. As a future professor at a PUI, any advice for working with undergraduates, and how do you pick projects?
You know, I think you can take a look at what's available to you. It's very institution dependent, but Hope's a pretty research rich place. We're really blessed with some good instrumentation. And I really do think I can do the same thing at Hope that I could do at MIT or the ETH, but it's going to take a lot longer. So you got to have an eye to not being scooped. You got to have a unique idea. You got to have something that's that interstitial space where you can find something that you can work away at a while. And what do I mean by slower? So if you picture what could you assume, not that you changed gears entirely for your postdoc, but picked a postdoc that you could just go at hammer and tongs. You had the skill set for it already. If you had a postdoc like that, where you were already well equipped with all the techniques, you didn't really need to learn much new, but you're tackling a new project. If that describes your postdoc experience, what you can get done in 1 year by yourself is about what you'll get done in 5 years with undergrads by the time you come up for tenure. And so that's a really good rule of thumb. So you need to think along those kind of scales. Maybe it means publishing more communications and less massive papers, right? I worked for my PhD was with a guy that tended to publish 20 and 30 page JAX monoliths. I need to think about, okay, what's a more discrete publishable unit here? But really, you can do all kinds of things, and you should just think about what's the instrumentation I have available to me, and how do I find something that's just enough different that, George Whiteside's at Harvard isn't going to do the same thing and knock out in 6 weeks what's going to take me 6 years.
Enough. Second question here: are these dyes mechanically sensitive? Can you put them in polymers and stretch them to cause color switching, or even just hit them for color changes?
You know what? You make me want to go into my lab and bang these with a hammer. I have no idea about that second part of the question. The first part of the question is really cool. And so some people, not with these ones, but with conventional azo dyes, people have put them between AFM probes and done single molecule mechanical stretching, and that's known to work. I'm not sure. Some photomechanical materials, I don't know if they're azo based. I have seen where they put them into polymer and stretch the polymer, and you can see some of the color change too.
That's cool.
That's an area that I'm much less familiar with, but it's really cool. I've got a colleague who's into what's called tribochemistry, which is kind of the chemistry of friction. A guy named Jeff Rack at University of New Mexico does some things along these lines too. He calls his photonastic materials, is another name for photomechanical materials. So there's a lot of cool stuff out there, but you could imagine you'd have to make them awfully darn cheap if you're going to use it to make your Amazon packaging, so you know whether or not somebody dropped your Xbox before they put it on your front porch. But that would be a cool thing to do with these.
Yeah, absolutely. So I'll ask along the same lines. How stable are these dyes that you've made, and in terms of like, can you do the switching back and forth, repeatedly, that kind of thing?
Yeah. So that's sort of their dirty little secret. Yes is the answer. They're really, really great and good and stable as long as you protect them from something. So you can sit them out at room temperature on the bench, open to air for months or years, as long as it's dark. And you can photolyze them back and forth as much as you want, as long as you do it under carefully anoxic conditions. But you need one or the other. If you photolyze back and forth in the presence of oxygen, you'll decompose them after a couple cycles. But if you scrupulously degas them, then you can drive them back and forth as many times as you would care to. And Abrahamian's papers, those two Jack's references I gave, he does that many times, and we've replicated that ourselves. Again, you just got to bubble it with argon really, really well. We know when, and that's sort of a good litmus test for how up to speed a student is whether or not they're able to make these nice and reversible, or if they kind of crap out after a couple cycles. Now people say, wait, so is that any good at all? Because, isn't everything going to be an oxygen in real life? And what I come back around is two things. Number one, you're putting them in a plastic. Most plastics aren't that oxygen permeable. That'll help. More importantly, we said that these are going to be of most use either transdermally. I don't know what the oxygen concentration in tissue is going to be, or in outer space or at high altitude. So, right, the NASA applications, the Air Force applications, they probably don't care too much. I'm not sure about the transdermal applications, but the polymer itself will help protect it from oxygen as well.
Absolutely. I think you're exactly right there. Yeah, it may not last forever, but doesn't need to necessarily either.
Right.
Well, fantastic. I think that's all the questions we've got. So I'll thank you again, Jason, for a fantastic talk. It's really cool work that you're doing, and it's like you said, you're you're doing all the same stuff at a PUI that you could do anywhere else, and it's really interesting work that you're doing. So thank you for sharing that with us and letting us see all of it.
Fantastic, Ben. Yeah, thanks so much. I appreciate you and I appreciate your audience. This was a great time. I thank you for the invite.
Yeah, absolutely. Let me get you to hang around for just 1 second, and we'll talk just briefly, and then I'll let you go. So for everybody else, we will be back at about 7 o'clock for our next talk. So I hope you'll come out and join us for that. In the meantime, maybe, depending on where you are, maybe grab an early dinner or something. And we'll see you at 7 o'clock.